Hello everybody, these are the notes for 10.4 on transcription. Transcription is the first step in making proteins out of the DNA. Today we are going to explain how DNA controls the activities of the cell, describe the three steps of transcription, and explain the role of the Tata box. There are two processes that go on in order to make a protein. The first of which is transcription, which is the process of making that mRNA. The mRNA will then be a template for translation, where we translate from the language of nucleotides into the language of amino acids, which make up proteins. The genetic code is the rule. And this rule states that a sequence of 3 nucleotides, or 3 letters, code for 1 amino acid. Again, these amino acids link together to make up proteins. Let's start with transcription. This is the making of mRNA. Actually, this is the making of just about all 3 of the types of RNA. This is the same process that makes tRNA and rRNA. So in transcription, here's an overview. Our pink strand represents our DNA. The blue strand represents the mRNA strand that is being made. This big gray spaceship looking thing is our RNA polymerase, and it is an enzyme. The DNA polymerase attaches to the DNA. It starts to unwind the DNA, and then it uses this strand as a template to make the mRNA out of nucleotide bases. The mRNA comes out the bottom here as a single strand, and the DNA rewinds itself up as a double helix. Step 1 is initiation. This is how we're going to start the process of transcription. RNA polymerase, along with some other transcription factors, bind to the DNA at a promoter site. The Tata box is simply this promoter sequence. The TA nucleotides are what allow the RNA polymerase to bind and tells it where to begin transcription. Here's a simplified picture. The blue strands here are the double-stranded DNA. This guitar pick looking thing is our RNA polymerase. The green promoter region is where we would find the TA code. Step 2 is elongation. This is where the individual nucleotides are added to create the long strand of RNA. Remember, RNA nucleotides replace the T with a U. Remember, there is no T in RNA. Here's a picture of Step 2, elongation. Inside the RNA polymerase, this very large, still somewhat looks like a guitar pick, the DNA is unwound, so that's shown here. The template strand is shown in dark blue. The other strand is light blue, and they come back together at the other end of the RNA polymerase and twist back up into the normal double helix shape. In the middle, while it's unzipped, the RNA polymerase will take RNA nucleotides, which are represented in red, and create a complementary strand of RNA. As the DNA rewinds together, the mRNA is released from the RNA polymerase in a long chain. This continues, in this case, in this direction, what we refer to as downstream. So it heads downstream until it reaches a stopping point. Termination is the stopping point. We actually call it a termination signal. Once the RNA polymerase travels down, here's our promoter region, our TATA box. It travels down the DNA, unzipping it and creating the mRNA. When it reaches the termination signal, the RNA polymerase simply falls off, releases the mRNA strand, and it's free to bind on another section of DNA and start the process all over again. Our completed RNA strand, the mRNA, will head out of the nucleus into the cytoplasm, where it can begin the next phase of making a protein. There are 2 videos. They are located on my YouTube channel, and if you'd like to look them up, that would be great. They will help give a good visual of what's going on.